My name is James Gaffney. I'm a senior in animal science. My project is investigating the potential of plant-derived molecules for controlling multi-drug resistant Acinetobacter baumannii. Acinetobacter species are bacteria that are commonly found in the environment, but A. baumannii is a little bit different. The natural host seems to be human beings. What we've been finding is that A. baumannii is nosocomially transmitted, that is, it's moved through healthcare facilities. We've been looking at, because it is a multi-drug resistant organism, different molecules from plants as a means of controlling A. baumannii. Conventional antibiotics, the organism is resistant to 8, 9, or 10 different classes, so we're looking for something that could potentially be used as an alternative or an adjunct to conventional therapy.
Where are these bacteria coming from?
In some cases they're coming from non-U.S. patients in military hospitals in Afghanistan and Iraq. A number of studies have been done looking at the epidemiology of Acinetobacter baumannii. But we've been finding is that a lot of it is persistent in the hospital setting, that is, sitting on catheters, ventilators, and other areas, because A. baumannii forms what are called biofilms. They're extracellular polysaccharide matrix that helps in adhesion as well as resisting disinfectants and other drying factors.
So what did your research determine?
So looking at this project, I was investigating, I had two main objectives. I was looking for the minimum bactericidal concentration, that is the minimum amount of these molecules that would be inhibiting growth of A. baumannii after 24 hours. In addition, also looking at the minimum bactericidal concentration, which is preventing the growth entirely after 24 hours, that is a 6-log reduction in growth. I was also looking at the time kill assay, doing a bactericidal kinetics experiment to see, if these molecules are inhibited or completely killed, at what time over 24 hours these effects are being exerted. These images will actually give you a very good idea of what we found. We did find that the molecules were effective. Looking at trans-cinnamaldehyde, which is from cinnamon, over 24 hours, the organism was controlled at the MIC, the minimum inhibitory concentration, in purple. And just a slight bit more, at 16 millimolar, the organism was significantly reduced by 6 hours and actually killed by 24. Which was a very interesting result. We found similar effects for carvacrol, which is from oregano, as well as eugenol from cloves. What's interesting is that the concentrations that we're using are in millimolar, so adding maybe 5 or 10 microliters to a 10 ml tube are having significant effects. Some of the directions that this research should be taken, this is obviously in vitro. We're not using an animal or a human model yet. But we've been looking towards maybe using these molecules for controlling A. baumannii in future projects.
What did the private support mean for your research?
It's important for us to have media in the lab. For me, I'm making plates every day. I'm making tubes. I need inoculating loops. Running a lab is very, very expensive, particularly in the hard sciences where you have definite needs, especially in microbiology where you're constantly turning media over. Having that extra support was very important to me, as well as the ability to live up here for the summer and conduct my research knowing that I wouldn't have to work on my own and to support myself.